All right, so thanks. Yeah, I'm Drew Blount. I am just a software programmer at Wildme in Portland, Oregon. And unlike a lot of you here, I'm not going to be presenting a particular research project, but more sort of giving an overview of who we are, what we do, a pretty good follow-up to the last talk, and what we're all about. So first, just for some context, I'd like to explain to people who may not be aware what a mark recapture study is. So in wildlife biology and ecology, it's a very important type of study where you go out, trap a wild animal, don't hurt it, maybe put a tag on its ear, and then in the future you can recite that animal and collect data about that one individual. These studies are also used to find accurate population estimates and other things like that. And what Wildbook is basically a tool to enable photo ID mark recapture studies. So photo ID mark recapture has become very popular with the advent of computer vision. You don't have to actually trap the animal and put a tag on its ear if you can get a computer to automatically identify photos of that individual animal. So a particular zebra might be recognized in photos. So I'm just going to give an overview of Wildbook, talk about our organizational model, which I also think might be of interest to some of you folks, and talk about the goals and challenges going forward. So first, rather than do a live software demo, here's just a demo via screenshot. So here is Flukebook. This is one of our many Wildbooks. We have multiple different deployments of this piece of software. And fundamentally, it's a web app. It's a web app where you can go and upload batch data that you've gathered, upload sort of incident by incident or encounter by encounter data. Flukebook is a website where people collect data on gray whales, humpback whales, and sperm whales. And here's some of the other pages on Flukebook, just to give you a feel for the site. So here's just a list of all the marked individuals we have. Looks like the thumbnails didn't load here, but we have over 11,000 whales have been identified on Flukebook, and that's all over the world, in the middle of the oceans in some places, and just off the coast in others. Here is some information we have on just a single individual's page. So you can see all the times that that individual has been seen. This one was encountered 7 times, you can see there. And you can go through and look at all the pictures of that whale, see the different times it was photographed. And here is some interesting map information. We have stuff from 2 different Wildbooks here. So this is actually from Whalesharkorg, which is a thing that's just like Flukebook, but for whale sharks exclusively. And this map shows different places where we've cited the same individual. So one interesting conclusion that I think actually produced a couple published papers out of Whalesharkorg was that there's a lot of whale shark populations that sort of have 2 different homes. And you can see that's about the length of Cuba there, but the whale shark sort of hangs out in those 2 areas. And these sightings are sometimes multiple years apart and very far apart from each other. So you can imagine it'd be very difficult for one research team to have tracked this whale. It's only possible because there's collaboration of different people collecting data in different places, uploading it to the same database, and the computer vision recognizing that it's the same individual. On the right here, this is a visualization we have from one of our projects with zebras. And here we have 2 different species depicted on the map. There's plains zebras and Grevy zebras. And these types of things are produced automatically within Wildbook. So a big goal of Wildbook is to not only gather all this data that people have collected throughout the world, but to automatically provide tools for analysis for ecologists to use. So here's a picture of Whaleshark.org I mentioned a second ago. Another main goal of Wildbook is to facilitate collaboration between researchers and citizen scientists. So when you scroll down on our homepage, you see information about the people that have contributed here. Simon's one of the directors of Wildme. You can see folks who have spotted recently. I think that this guy on the top is a tour guide that gives scuba tours. And we collaborate with him because when people go out on a tour to try and see whale sharks, they pay money, they're going on this expensive vacation, and now they get to feel like, I'm a scientist collecting real data. So people really like that. And sort of community engagement is a really important thing for us. Again, these numbers on the bottom I kind of like. We just have a whole bunch of whale sharks that have been identified and almost 35,000 different particular encounters in our database. Further on that community engagement thing, here's a Wildbook we set up for the WWF in Finland studying ringed seals. This is our footer on that web page. And it says here, what can I do to help adopt this seal? So hopefully that seal on the right will raise a lot of money for the WWF because people like to, this adoption service we offer gives you, you can choose a particular individual that's been identified, and then you'll be notified via email anytime it's recited. So maybe you go on a tour, you find a whale shark, you think that's cool, and then now you might get an email 2 years later, hey, we found that whale shark on the other side of the ocean. And then you can look at the pictures and do stuff like that. So just to sort of summarize a little overview here, Wildbook is a tool for collaborating between researchers. It's a tool for just simply interpreting data, doing data analysis, and getting valuable information about the populations and individuals being studied. It can help conservation by improving community engagement, and also, of course, the data that's gathered could be important for those decisions. And it brings the public into research at scale. So a big goal with Wildbook is to really broaden the scope of a lot of projects that previously had been done by independent research groups that were not necessarily collaborating. Maybe they would meet at a conference and find out that they're doing similar things. But our goal is to sort of get as many people under the same umbrella as possible if they're working on the same type of studies so that they can share their expertise and so that they can increase the scope of their research. And like the maps I showed you earlier, Wildbook allows us to answer a lot of interesting scientific questions. So, for example, are zebra stripes genetic? Does a foal look particularly like its mother in terms of stripes? That's a question that you couldn't answer previously without mass individual tracking. Other things that you can find out very readily with Wildbook are co-occurrence of individuals. So we've tracked a large zebra population in the Lewa Wildlife Preserve in Kenya. And one thing that zebras do is they travel in harem groups where there's one male and multiple females. So one question we're hoping to answer in the next few months is, so sometimes these harem groups will combine into a larger herd. Not always, but there will be maybe 3 harems together. And the researchers there are wondering, what's the relationship between those males that are leading those harems that are willing to collaborate with each other? Because usually the males are quite competitive. So we might be able to find out, well, when males are willing to combine their harems, that's because those males grew up together. Or maybe when they were young, they were seen in the same bachelor group very often, so they're familiar with each other. And these types of things just appear on web pages when you go there, and it's hopefully so much easier than sort of designing a study from scratch around it. And another thing we can see, of course, is the long-term trends of the population. This allows for a really totalized view of all the animals across a landscape. So we can see their range and understand when things are out of the ordinary. So I'll just list through a couple different deployments we have of Wildbook here. So we have Whaleshark.org, which you saw. There's Flukebook. There's Manta Matcher for manta rays. We have Spot-A-Shark, which does gray nurse sharks. We have Project for Zebras that are not public-facing and just used by researchers. And we're building the Internet of Turtles, which Tanya mentioned in a talk yesterday you may have heard of, and the ringed seals for the WWF. We're also working on projects with elephants, wild dogs, sunfish, and many other species that I didn't remember to put here. So our organizational model is, I think, like a lot of projects that kind of spin out of academia, is sort of a research project that outgrew the scope of any one grant. So we want to provide this collaborative platform to people, do it as a nonprofit. And something that's really important to our entire model is that the code is open source. So on the one hand, there's the just feel-good philosophical side of having open source code, and we're not, collecting anyone's data and keeping it from them or anything like that. But what's really important is that when we have multiple different research teams using the same tool and it's open source, anyone can improve it. So, for example, a few years ago, someone said, why do you guys not have GIS support? We really need that. And then they built a tool, they had someone in their team that could understand our code, that made a GIS exporter. So then that tool is now shared by everyone that uses Wildbook, because it gets merged back into the main branch. So in that way, we're not just collaborating in terms of multiple like whale shark researchers on opposite sides of the ocean, but you can have the whale shark researchers' software development also benefit the zebra researchers. So to just sort of illustrate this point with the open source code, I'll go through a little bit of the history of our computer vision that we did. So the first project that founded Wildbook was Whalesharkorg. Our founder went out and saw some whale sharks in the wild and learned about the mark recapture studies that they were doing back then, which involved physically tagging the whale shark with a little plastic thing that got stabbed into its side. And in those studies, the whale sharks, they would have to apply 100 tags to individuals to get to approximately recite one individual. So a lot of the tags fell off. They didn't have very good coverage of the population, and it just wasn't a very reliable way of doing research. So this was back around 2005 or so, 2003 maybe, before deep learning was a big thing, and we had pretty custom-built identifiers that used a lot of human domain knowledge for the identification task at hand. So here we used an algorithm that was developed at NASA for identifying constellations in photographs, because whale sharks have these really distinctive spots on their sides. And when you have two photos of the same whale shark, you can draw all the triangles possible between those spots in a given region. And then if the photos are of the same individual, there should be an affine transformation that makes all the triangles line up with each other when you transform. But that won't work if it's a different individual. So this doesn't work on zebras, of course, but it does work on spotted sharks. And it does work on spotted grouper that live off the coast of California. So the fact that this is already deployed in an open source tool means that those other species can just start right off the bat. Basically download whaleshark.org and just upload pictures of spotted sharks this time. Here's another case of a very domain-specific matching algorithm we used. The trailing edge of humpback whale flukes is very distinctive. And you can see that there's distinct sort of splotches and patterns on the fin, but the trailing edge itself is unique enough that we can match off of it. So we do a sort of extraction here of the trailing edge, and then used algorithm from digital signal processing, because we can approximate this as a function, and try and find different instances of that same signal in our database. And this algorithm is also applicable, we're hoping, we haven't found great results, but we think it could be used on elephant ears as well. And here is a stripe spotter, or a visualization of stripe spotter, which was an algorithm that was developed by a student in Tanya's lab that identifies zebras by their stripes, reads them like barcodes, sort of. Though now, what with deep learning and whatnot, we're moving towards a world with more general purpose artificial intelligence algorithms, certainly computer vision. And the algorithm we've been working on most recently with from Rensselaer Polytechnic is called Hotspot, and it uses deep convolutional neural networks to extract relevant features that are unique to the individuals in the photographs. And here's a turtle that we identified in two different photos, and you can see it finds regions of interest that are ellipsoids, and similar to the constellation mapping, there's just an affine transformation from the ellipses in one image to those in the other that is much more similar between photos of one individual than photos of two different turtles. So Hotspot is where a lot of our focus is these days during R&D, because this has a lot more potential to be applied to many more species. Like the trailing edge algorithm for humpback whales is very specific, and this is, something that we can apply to anything that has good stripes or spots. So what we're developing now is, of course, a computer vision can always be improved. One of the things that we are really interested in is processing huge batches at once. So we'd like to be at a point where someone could upload a folder with 10,000 files, or 10,000 images, some of which may or may not have animals in them, and then automatically detect the species in each photograph, and upload it to our database. So there's always plenty of room for improvement there. We can always broaden the scope of the ecological studies that are involved, and at any point, several different research teams using Wildbook are probably working on things like this. So automatic population tracking and estimation and things like that are in the pipeline. And a really big goal of ours in the near future is to start combing the internet for images of animals. So we've started doing that and have had pretty good success just matching random Google image search pictures of humpback whales. So that's potentially a huge boon to researchers who, so for whale scientists, a big chunk of their budget is just going out whaling all the time. And there's, of course, millions of photos on the internet that might be of value to researchers. And let's see, so just some of the challenges we have are security, of course. So if we do build an elephant Wildbook, the international ivory trade is like many billions of dollars. And if you have a database that contains an entire population of elephants in it and all the locations they've ever been cited, that's clearly a pretty big target. So security is very important, which is going to be the topic of Jim Basney's talk in the next section. And since we're so focused on collaboration, we found that different academic communities have different attitudes towards that. Some biologists are very eager to share their data, and some biologists, whale biologists in particular, are very sort of invested in the data sets that they have collected. It's sort of an asset for them to get grants and to, get tenure and so on, and they're afraid that if they share it, other people might beat them to publication. So there's definitely an issue with convincing people that they have more to gain from collaboration than they stand to lose. And as I think we heard a couple times yesterday, I put in bold here, because me as the sort of youngest software developer on the team, I'm always the person that has to grapple with this the most. But every research team will, in terms of the teams we've worked with at least, they just invent their own data models from scratch. And it always makes sense for them. They're collecting the data that they need, but you'll end up with two teams of zebra researchers that are working on other sides of the same conservancy. And they just have completely incompatible data models sometimes. So a big issue is finding the sweet spot between having a core set of features that are shared between all Wildbooks for interoperability and sort of totally customizing to one research team and use case. So I think that's the bulk of my talk. So maybe there'll be some questions, and feel free to email me or visit wildme.org. There are too many people to explicitly give everyone credit here. Thanks.
So, I mean, it's a question I ask Tanya all the time too. Are you collecting data that would help us understand the sampling biases of the observation process behind these? Because that's a big issue, obviously, in interpreting this data.
Yeah, and certainly with tourists, where there's even a bias of cute animals.
I mean, that's lovely. We know exactly. It's like he's walking the same transect every week.
We don't have any tools for that right now, and I know that Tanya is working on it as an active research question because it's a bit of an unsolved problem. But it's certainly a big issue. It's very different. Some Wildbooks are very driven by citizen science and these tours and whatnot, whereas the zebra Wildbooks we have will have trained research assistants that are going out on patrols every day that are sort of deliberately reducing their bias as much as they can. But at this point, all that we have is sort of this heuristic answer to that question.
Is this nonprofit focused on this vision-based applications software, or is there room for other kinds of software?
There's definitely room for other types. For example, we store information about samples that might have been gathered when you view an encounter. Fundamentally, you could absolutely have a Wildbook that doesn't have any photographs in it. But it is very tied to the model of mark recapture studies, of tracking individual animals and particular populations.